Thank you very much for giving us the opportunity to present our project. We are PixelQuality, and we develop best practices for publishing images. Scientific research is grounded in data, and images are one source of this data. In biomedical imaging, with microscopy is a powerful tool that allows us to capture stunning biological processes. We can take a deep look into cells and study fundamental biological principles. We can look at tissues and how they change under disease conditions, here with a histopathology image of a skin cancer. And we can study entire organs and even entire organisms, here represented by a fluorescent turtle embryo. We can also study very small things, here represented as a coronavirus imaged with electron microscopy. We can image objects in their entirety with 3D imaging, here shown on a Drosophila ovary. And we can image very dynamic processes, here shown on a living, beating zebrafish heart. These images are compelling evidence in scientific publications. However, many things can go wrong when preparing images for publication. For instance, they can be processed incorrectly. Here, I show you the image of a cell with the nucleus in magenta, the cell outline in white, and cell organelles, lysosomes in green. This could be a typical research result. The researchers would count the lysosomes, they would study their size and the distribution within the cell. The scientists need to process these images in order to visualize them in a publication. These are simple intensity adjustments, for instance. These are important and necessary steps towards visualization, but this processing can go wrong. Here I show you the same image with the same information with different processing settings. There are more lysosomes visible now, they are bigger in size, and they are distributed all over the cell. Currently, there are no guidelines that mandate to document such image processing settings. Images that go into image figures are not shared accessible. Therefore, they are not accessible for review. And method sections are often sparse in the performed image processing and analysis. And another important issue is legibility and accessibility of the image result in the images. Here, my colleague Helena Jambor made an important observation. She studied many publications and reviewed the images that are in these publications, even from high-impact journals. She discovered that only 10 to 20% of surveyed images are fully understandable. What does she mean? Many images are not colorblind safe, therefore not accessible for all audiences. Often scale bar, really basic annotations are missing. Props are not annotated in their origins, and annotations explaining what is shown in the images are not on the image. Also, explanations are missing or are incomplete in figure legends. All of this degrades the scientific results and makes the information depicted in images inaccessible for other scientists. We believe there's a lack of unified guidance of how effective images should look like in publications. And there's a lack of training of scientists to achieve that. In order to address this widespread research quality problem, we founded a working group within the organization Quality and Reproducibility in Microscopy. We have now over 140 members from all over the world. These represent scientists from many different research organizations, such as the Max Planck Society in Germany, Harvard, even NASA joined us. We also have scientists from joining us from companies, for instance, from microscopy companies such as Zeiss. And also we have collaborators that are scientific editors. With this community, we were able to develop guidelines that help for the publication of images and image analysis. We summarize these guidelines in easy-to-use checklists that should enable authors, reviewers, and editors alike to improve the quality of published images. We were published last year in Nature Methods, and we were well received by the scientific community. So they are interested in this content. We have over 30 citations and over 33,000 accesses online to date, making us the #1 Nature Methods article of similar age. Now, our next challenge is to make these guidelines common scientific practice. With support from the Einstein Foundation, we believe that we can realize the full potential of these checklists. We want to create open source training material. We want to disseminate them widely among scientists, and we want to make them up to date to current usages of artificial intelligence. How are we doing this? So our existing global team is currently developing training material. We develop them iteratively in cycles of open review, and we will involve key stakeholders, communities from imaging, microscopy, and research. This development is fully in the open, and the material will be open source. Funding from Einstein would allow us to accelerate this process greatly by holding workshops in order to develop broad material covering diverse perspectives. All the content will be released and collected on our website, where a first version is already online. Next, we want to widely disseminate our guidelines and to develop training material. In the past, we have been doing this piecewise via courses, scientific talks at conferences. The targeted specifically towards scientists, facility staff directly. This is a very rewarding process, and we received fantastic feedback here. However, we want to scale this process up. We want to now target microscopy facilities and large research organizations, which we already collaborate with. We want to hold train-the-trainer workshops and co-develop their course content, which will ensure widespread and sustainable dissemination to many researchers. We also aim to collaborate with publishers. Here we can show first results with a collaboration with Springer Nature. Springer Nature is one of the largest scientific publishing houses with over 3,000 journals. This includes 290 peer-reviewed journals in biology, medicine, and health. There is a trial adoption of our guidelines with a subset of the Nature portfolio journals. The ultimate goal is to uptake them across all Springer Nature journals. But we can take this further. Finally, the last years have shown us the great potential of artificial intelligence to accelerate the scientific process. I show you this on one example of impact of a machine learning method. Here, I show you a flatworm imaged with microscopy. Living organisms pose a specific challenge for microscopy, as we have to image them with low light, otherwise we kill the animal in the process. This leads to low signal images that are very noisy, here shown on the left. You can see also other imaging artifacts that are unavoidable in this process. Now, with applying machine learning methods, we can restore these low signal images to a high signal images. But how can we make sure that the results are trustworthy? We need guidelines that give scientists and reviewers clear rules of what is and is not permissible in publishing such images. And we need to give them guidance on how to document these transparently. For instance, by clearly annotating such processing in the figures, or by supplying metrics that give a certainty towards the impact of such an AI model. What we want to do is to apply or establish the process to this research quality problem. We will review the current status in publications. We will address different affected communities to form a strong team, which will then expand the existing guidelines to establish best practices for using AI in processing and generating images. I showed you now with that PixelQuality has identified an important gap in research quality, specifically with the lack of quality of published images. I showed you how we took the first steps in addressing this quality problem with our community-developed guidelines. Now we need your support to take the next steps in creating effective and open source training material, disseminate them to a large scientific audience, and expanding the guidelines through AI. I want to thank my colleague, Helena Jambor. She's also available for a Q&A. I want to thank my team, the PixelQuality team, who with their support and enthusiasm make this work possible. I want to thank Plimi as an organization and our research institutes that host us, which allowed us to do this on our side. And with this, I want to thank you for your attention also, and I'm open to receive your questions.
Thank you very much for stepping in on very short notice, and please accept my apologies because I didn't introduce you. So before we get into the discussion, I will introduce you post hoc. So Christopher Schmied is a senior research data scientist at the Leibniz-Institute for Molecular Pharmacology. He's also the lead data scientist at EU Open Screen, a non-profit European research infrastructure consortium. Christopher's research focuses on the analysis, processing, acquisition, storage, and management of scientific image data. And together with his team member, Helena Jambor, a specialist in biological and medical data visualization, currently an associate professor at Chur University of Applied Sciences. He has, as we've heard, actually established guidelines for clear image publication in microscopy, which aim at enhancing reproducibility. So apologies and thanks again. And we now have time for discussing this project. And I know that maybe Helen and Susie could start it off. So please just kick in, whoever wants to